Hey everybody. The next thing we're learning about is some more detail about how proteins are made. So remember, the two processes in protein synthesis are transcription and translation. So before we dive into some detail, one important thing to remember is that it's enzymes that are controlling most of the processes here. Remember, enzymes are a type of protein that causes a chemical reaction to happen. So it's enzymes plus a few other proteins that are making all this stuff happen. Okay, so the first thing we'll look at is right back at the DNA in the nucleus. So here is one gene. So this region here is our gene. And remember, the gene is like the recipe. It's the instructions for making a protein. So once that gene is ready to be expressed by the cell, the chromosome, well actually the chromatin, has to unravel a little bit. So remember back when we learnt about DNA packaging, chromosomes have the DNA, but they're also wrapped around these proteins. So all these supercoils of the chromatin, they have to unravel a bit until they expose the DNA strand where the gene is. And again, that's controlled by proteins. So the first stage is the chromatin unwinds. Okay, the next stage is the two strands of the DNA separate so that an enzyme can actually get in. So remember, DNA is made of two strands, but only one of those strands is needed to be copied. So the DNA strands separate. And then, as you can see here, one of those strands of DNA is used to make a copy. So that's step 3 in here. The strand that's used to make a copy is called the template strand. And that's the code that's then copied onto mRNA. And remember, mRNA is like just a photocopy of the gene that gets taken to the ribosomes later on. So the DNA sequence of the gene is copied onto RNA. And then once the whole gene has been copied, the RNA breaks off and we call it mRNA, or messenger RNA. So here's our mRNA molecule, and now it has to go and find a ribosome to actually make a protein. So this is like, if we're going back to our cookbook analogy, this is the person holding the copy of the recipe, and they need to take it to the kitchen to make the cake. So here we are zooming out. Here's the whole cell. We've done transcription. We've got our mRNA, and the mRNA has to pass through the nuclear membrane of the cell, out of the nucleus, and into the cytoplasm. So that's the next step. That mRNA travels out of the cell into the cytoplasm and it attaches to a ribosome. And a ribosome is the organelle that's going to make the actual protein. So here's an example of showing that here. So we've done transcription, we've made our mRNA. Now we're actually into translation here. So transcription ended after step 4. Step 5 is an in-between step. And here's translation. So translation is actually taking that code on the mRNA and making the protein. And the way it does it is based around these 3 base words, which are called codons. So there's a few things you need to know here. The first thing is that the genetic code, it's completely based around 3-letter words. Or in other words, it's like a 3-letter language. So every 3 bases on the messenger RNA have the code to attach 1 amino acid onto the protein. So you can see AUG, that represents, or that's the code for adding this particular amino acid. And then the next word along is AUC. And that corresponds to this particular amino acid. And then we have UCG. Then we have AUC, which is actually the same one again. And then we have GAU, and so on. So, amino acids, they're the building blocks for actually making the protein. The codons are a code for a particular amino acid, and they're a 3-letter code. And the final protein is made of all those amino acids joined together. So that is basically step 6 and the final step in making a protein. So you can see that cells start with that message on the DNA, and it eventually leads to making a protein at the very end, which eventually folds up to make the protein, and then it goes to the part of the cell it's needed and it goes and does its job.